Ben Beal. I'm an extension agent up at the University of Maryland in Southern Maryland. So today we're going to be going through the Integrated Weed Management Workshop. Today we're going to kind of go over herbicide resistance and talk a little bit about the history of it, how it came about, mechanisms of action, and creating effective herbicide plans.
All right, well, welcome everybody. As Ben mentioned, if you looked at your program earlier, this is an integrated weed management workshop. But this session, we're only pretty much going to focus on herbicides and herbicide resistance, which is the driving reason why we need to incorporate integrated weed management into our programs. So for this first part, I'm just going to go over herbicide resistance. What is it, and what are the different mechanisms that plants use that make them resistant to herbicides? So we can define resistance as a change in the sensitivity of a plant population to a particular herbicide. Herbicide resistance is the result of naturally occurring processes. Before a herbicide like glyphosate is even sprayed, there may be one individual that is a herbicide resistant biotype. But these individuals usually occur at very low frequencies. And what we do when we spray the same herbicide over and over again is we apply what's going to be referred to as a selection pressure. And if we use the same herbicides over and over, then eventually we're going to see more resistant biotypes. Over the years of using the same herbicide, we will effectively eliminate all of our susceptible biotypes, leaving only the resistant biotypes, which multiply, and then eventually we're only left with that herbicide resistant biotypes. However, lack of weed control doesn't always mean resistance is the issue. About 9 out of 10 herbicide failures are due to factors other than resistance. Many of these factors are the direct result of the application itself. For example, the herbicide may have been applied at an inadequate rate as the result of improper calibration, or not using the recommended rate. There may have been poor spray coverage, or in the case of soil applied herbicides, poor soil incorporation. And for example, contact herbicides such as glufosinate work better when applied at a greater amount of spray volume. The weeds may have been too large to spray with the desired herbicides. Remember, most labeled herbicide rates are based on weed size. In addition, weed size can also influence spray coverage, as larger plants may prevent the herbicide from reaching smaller plants. In addition, excessive dust can inactivate post-emergence herbicides. Herbicides like glyphosate and paraquat will bind to soil particles. So if your tractor is kicking up dust during a herbicide application, then that herbicide is binding to the dust rather than reaching its target. And problems can also arise from tank mix issues before spraying. So always be sure to read the label before mixing different herbicides, as problems can result from improper pH, antagonism, or just failure to use an adjuvant. Other factors are the result of soil and climate conditions during or after the application. Excessive wet soil can cause herbicides to leach through the soil profile, making them unavailable for plant uptake, and dry soil can prevent the herbicides from being activated or taken in by a plant. Seedbed conditions can influence weed seed germination. And as I mentioned before, herbicides can bind to soil particles. Soils with higher organic matter content generally tend to bind herbicides more readily. That is why application rates differ with soil type. And certain climatic conditions will also favor increased microbial activity, resulting in faster degradation of an herbicide. In general, microbial activity increases as the soil gets warmer to a point. And there may have not been enough of a rain-fast period for your post-emergence application. And the herbicide may have been just simply washed off that leaf surface. Then there's the issue of herbicide tolerance, which is the inherent ability of a species to survive and reproduce after a herbicide treatment. This implies that there was no selection pressure and this species was naturally tolerant. So herbicide tolerance is not synonymous with herbicide resistance. By definition, if a weed has never been controlled by an herbicide and there has been no change in the weed population's lack of response to that herbicide over time, then the population is tolerant. And tolerance is the basis for herbicide selection in many of our crop species. For example, if you look at the pictures on your screen, one of these species is tolerant to Roundup, and one of these species is resistant to Roundup. I'll give you a few seconds to determine which one. Well, if you guessed that the species on the left, the Morning Glory, was the tolerant species, you're correct. Roundup really hasn't been that good of a herbicide for Morning Glory. But if you guessed the species on the right, Palmer amaranth, was the resistant species, you are correct. Palmer amaranth populations throughout the country have been well documented to have glyphosate or Roundup resistance. So what can herbicide resistance look like in your field? Well, barring all of the factors I just mentioned, tolerance, soil conditions, spray conditions, etc., you may start to notice a few uncontrolled plants in your field after a herbicide is sprayed. However, after successive years, you spray the same herbicide and you see more and more of those plants are not controlled. But one of the key factors for seeing if you have herbicide resistance in the field is you'll see a bunch of surviving plants mixed in with dead plants. And there'll be no real pattern. If you saw a bunch of dead plants in a strip versus a bunch of live plants in another strip, you probably have a nozzle clogged. But in this case, for example, with horseweed, you see this entire field was sprayed with Roundup. But as you can see, there's a bunch of living plants intermixed with plants that were killed by the herbicide. So now let's shift gears a little bit and discuss the different types of herbicide resistance. Some weeds are only resistant to one herbicide. This is known as single herbicide resistance, while other weeds may be resistant to one or more herbicides, either through cross resistance or multiple resistance. Cross resistance occurs when a weed is resistant to two or more herbicide families within the same site of action. This is due to a single resistance mechanism. For example, a weed may be resistant to both Raptor and First Rate. Both are from different chemical families, but both inhibit the ALS enzyme in plants. Multiple resistance occurs when a weed is resistant to several herbicide groups with different mechanisms of action. This type of resistance may be the result of two or more resistance mechanisms within the same plant. The level of herbicide resistance may also vary within plant populations. The level of herbicide resistance found across all weeds and herbicide resistance mechanisms is not uniform. Rather, the level of herbicide resistance in weeds can vary by weed biology and the herbicide resistance mechanism. In some cases, resistance occurs when weed species survive applications at the labeled herbicide rate, while in other cases, a species may survive 1,000 times the labeled herbicide rate. And this awareness of varying levels of herbicide resistance is important for being able to identify resistance in the field. Weeds with high levels of herbicide resistance often don't show herbicide injury symptoms, while a weed with a low level of herbicide resistance may be injured, yet survives and reproduces. So let's look more at some of these mechanisms of herbicide resistance in detail. So these, again, herbicide resistance may occur through several different biological mechanisms, including an altered target site for the enzyme, overproduction of a target protein, compartmentalization or sequestration, or via enhanced metabolism. So the first mechanism of resistance that I'll discuss is altered target site. And as I mentioned the ALS enzyme before, and enzymes are substances produced by living organisms which act as catalysts to bring about some specific biological reaction. These enzymes are needed to synthesize these important molecules, such as amino acids, which are also important for growth and function. So here what we have is commonly described as a lock and key molecule. The enzyme substrate is the key, while the enzyme is the lock. And normally that substrate will fit into that enzyme lock or key very well and produce the amino acids the plant needs to live. Under cases of non-resistant plants, the herbicide actually will compete with that binding site for the substrate, preventing the substrate from binding, and therefore those amino acids are not produced and the plant dies. Altered site herbicide resistance occurs basically when we change that lock, and that herbicide is no longer able to bind to the enzyme, but the substrate can still bind to that enzyme and produce the amino acids the plant needs to live. Another type of herbicide, target site resistance was first noticed as being widespread early in the 70s and 80s with your group 5 herbicides, your triazines, and your group 2 herbicides, again, your ALS inhibiting herbicides. Target site resistance also confers a high level of resistance with weeds showing little or no response to the herbicide. For example, in this example with ALS resistant or group 2 resistant ragweed sprayed with Pursuit, notice that the ragweed has a very high level of herbicide resistance, and we really don't see that much injury until we spray at a rate over 100 times the field rate. And target site resistance can also confer different levels of cross resistance. I mentioned first rate or Pursuit earlier as an ALS inhibiting herbicide. It's a member of the imidazolinone family, or it's a group 2 herbicide. However, again, first rate is another group 2 herbicide, a member of a different chemical family, but if we have cross resistance, that plant is going to be resistant to both Pursuit and first rate. Another type of herbicide resistance occurs through gene amplification or overexpression of the target enzyme. Under normal circumstances, there are just enough of these enzyme target sites for the herbicide to bind and prevent those amino acids from being developed. However, what a herbicide-resistant plants will actually produce more of these target sites than the herbicide can actually bind to. Gene amplification also confers a high level of resistance with the weeds showing little or no response to that herbicide. However, to my knowledge, target or gene amplification has only been reported to glyphosate to date. If you notice from the example below, you have 2 susceptible populations that show the response you'd hope for spraying a weed with glyphosate. But if you notice the resistant population here in the back, you see almost no response from the untreated control up to, say, that around between the 1x and the 2x rates before you even start to notice injury symptoms. And even at the 64x rate, you see a little bit of green, and that plant will probably survive. Non-target site resistance is fairly new to us. This confers a low level of resistance in plants, and they may show a range of responses. In non-target site resistance, the plant can survive by limiting the amount of herbicide that reaches that particular enzyme or substrate target site. We will often see some response from resistant biotypes. For example, non-target site resistance may involve reduced translocation, which is simply the herbicide being translocated into the vacuole where it's again sequestered from that target site or broken down into that by that vacuole. Non-target site resistance can also be metabolic resistance. Now, metabolic resistance is what allows a weed to rapidly degrade an herbicide. This mechanism is why crops can be treated with different herbicide products and no herbicide injury symptoms occur. Metabolic resistance is also how we as humans can detoxify certain compounds. So metabolic resistance is typically a three-phase process. Phase one involves a slight modification of the herbicide, which predisposes it to further modification. Phase two involves combining that herbicide with a compound such as a sugar that facilitates the final step. And phase three occurs when that herbicide is transported into a vacuole or outside the intracellular space. Each of these phases is mediated by a particular enzyme. For example, the cytochrome P450 for phase one, GST for phase two, and ABC transporters for phase three. A lot of these enzymes are similar in humans. However, plants typically have multiple versions of these enzymes. For example, in humans, we have maybe 60 different versions of that cytochrome P450, while plants may have about 360 versions. And having multiple versions of these enzymes means the plant may be able to metabolize more than one type of herbicide. So how does that occur, and how does this metabolic resistance occur? Well, when applied at low doses, say a half-x of your labeled rate, individual plants can survive because they actually possess sufficient capacity to metabolize the herbicide. This leads us to the phenomenon of creeping resistance. These lower herbicide dose rate application rates to allow plants with minor resistance genes to survive, providing the genetic material for future herbicide-resistant biotypes, which can be enriched through cross-pollination within the population. For example, a study out of Australia found that using reduced rates of holon resulted in biotypes that were 6 to 50 times more resistant within 3 generations. And these lower herbicide rates may be attributed to deliberate rate cutting, antagonistic mixtures, spraying of older or larger plants, spraying when the plants are stressed, or simply non-uniformity of an application. Multiple resistance can also occur when certain enzymes, such as cytochrome P450, display increased metabolism. This not only allows a biotype to metabolize one herbicide group, but multiple herbicide groups as well. For example, a water hemp population in Illinois, researchers previously reported that it was resistant to the group 27 herbicides, as well as resistant to 2,4-D. Group 27, or the HPPD bleaching herbicide resistance in water hemp, is due to increased activity of that cytochrome P450 enzyme. Esmetolachlor and 2,4-D resistance in other weed species has also been linked to increased activity of this cytochrome P450 enzyme. So it's possible, if not likely, that the genes providing resistance to the group 27 herbicides are also responsible for new resistance in this recently identified population. So again, metabolic resistance can confer resistance to different chemical group, allowing for multiple modes of action and possibly sites of action not yet discovered. So rotating different sites of action may not be enough when you have an enzyme like P450 that controls these multiple metabolic resistance processes. Thus, metabolic-based herbicide resistance is becoming a major threat, as it can automatically confer resistance to existing and new or yet to be discovered herbicides. Originally, resistance to site of action was thought to be low risk when talking about metabolic resistance in cells, but we are seeing more and more examples of metabolic resistance increasing. For example, with group 15 resistance in Palmer amaranth, and again, group 4 resistance in pigweed species as well. However, metabolic resistance is one of these things that's difficult to study just simply due to the complex families of these different enzymes like cytochrome P450. A high genetic diversity of different weed species, especially with your crossed and pollinated species, and just simply complex genetic controls of metabolic resistance. So those were the different ways herbicides can be resistant. So let's talk about what we've done to kind of promote herbicide resistance and how we got here. Now, the first thing and the most obvious thing is poor herbicide management. And when we talk about poor herbicide management, we're primarily talking about reliance on a single site of action or a single herbicide group number. For example, what we saw earlier in the, like I mentioned, the 60s or 70s and 80s with target site resistance and the group 2 or ALS inhibiting herbicides. After Roundup Ready crops developed, we saw more resistance due to gene amplification with group 9 resistance or glyphosate. And when we've been seeing where we've used ineffective herbicide rates, we've also developed metabolic resistance to herbicides such as esmetolachlor. Next thing about herbicide resistance and how we got here is we simply failed to control weed escapes. We just ignored them. For example, you may have just one single Palmer amaranth plant remaining in your field at the end of the season. But as you'll find out later in this workshop, Palmer amaranth has very prolific seed production. So that one plant, you spray it with Roundup and it doesn't die. Now, don't worry about it. However, next year, that one plant produced seed and you have even more Palmer amaranth in your field. The following year, you allow those plants to produce seed, and this is what you get. And I challenge you to actually find a crop to harvest in all of that mess. And the last part of how we got here with herbicide resistance is we failed to prevent the transport of these escaped weeds. Now remember, weed seeds can be dispersed by several mechanisms. They can be dispersed by the wind, by animals, either by eating them or just by transporting them on their fur. It can be transported by water. But we also have to remember is we live in a global economy. So, weed seeds may simply be blown off the back of a truck. You can also transport herbicide-resistant weed seeds by harvesting an infested field and then moving your combine to another field without cleaning it. So our goals for this workshop are, for one thing, to help you become familiar with herbicide resistance issues in the region. Next, we're going to help you design a herbicide spray program that will help us to conserve the available sites of action that are still effective against these resistant weeds. And finally, how to incorporate non-chemical tactics to manage these herbicide-resistant weeds. Thank you all.
We did want to acknowledge support for this project, particularly the Maryland Soybean Board, as well as throughout the Mid-Atlantic, University of Delaware, Virginia Cooperative Extension, University of Maryland,